So thanks everyone. So the next presentation is on updates on soft tissue neoplasms of uncertain differentiation. And as Prof mentioned, the principal way of classifying soft tissue tumours is by line of differentiation. But there is a large group of tumours of uncertain differentiation which do not resemble any specific cell or tissue or structure in the body. And these are a very heterogeneous group of neoplasms. This is the group here. And now we understand that many of these are defined by characteristic genetic events. In red, we have highlighted the new entities in this group, the NTRK rearranged spindle cell neoplasm, so an emerging group of tumours unified by the presence of various NTRK gene rearrangements, and then the group of undifferentiated sarcomas, which I've included as a new entity because in the previous edition of the WHO classification, these represented a standalone section, and now they have been subsumed into the group of tumours of uncertain differentiation. So many of these will have been discussed by Cyril or will be discussed by him or me in later presentations. So I'll just focus in this talk on the undifferentiated sarcomas, as well as a small number of other sarcomas not mentioned in the other talks, ossifying fibromyxoid tumor, extraskeletal myxoid chondrosarcoma, and intimal sarcoma. So we'll start first on this group of undifferentiated sarcomas, which comprises the largest group and is an extremely heterogeneous group. So these tumours, undifferentiated sarcomas, are those in which there are no specific defining features other than the lack of any identifiable line of differentiation using current available technologies. And we can divide these groups, this group morphologically, into four morphologic types. So undifferentiated pleomorphic sarcoma, and this is what most of us understand by undifferentiated sarcoma. So we think of undifferentiated pleomorphic sarcoma, UPS. But there are three other morphologic patterns: undifferentiated epithelioid sarcomas, undifferentiated spindle cell, and undifferentiated round cell. So the undifferentiated epithelioid, spindle cell, and round cell tumours will be covered by Cyril or me in coming talks, but we'll focus in this one on pleomorphic sarcomas, so undifferentiated pleomorphic sarcomas. This is by far the largest group of soft tissue sarcomas. Approximately 2/3 of all soft tissue sarcomas are classified as undifferentiated pleomorphic sarcoma, UPS. 75% of these are highly aggressive, which is a little bit interesting because we'd think that 100% of these will be highly aggressive tumours. So we'll come to that later. And so let's talk briefly about the concept of malignant fibrous histiocytoma. So what we now know as undifferentiated pleomorphic sarcoma was previously known as malignant fibrous histiocytoma, and we'll talk a little about the history. So malignant fibrous histiocytoma was an entity that was coined by authors in the past due to because of these markedly atypical cells in fascicles in malignant neoplasms, and they were thought to have this fibrohistiocytic lineage. And it was thought that these were modified fibroblasts which assumed phagocytic properties in tissue culture. So these were thought to represent a specific lineage of neoplasms, and these gained this concept of MFH gained widespread acceptance as an entity. And by the 1980s, the concept of MFH was actually the largest category of sarcomas. So MFH represented the most common sarcoma subtype. But after that, in the late 70s and 80s, particularly with the advent of immunohistochemistry, as well as the refinement of electron microscopy, it was discovered that there was no actual evidence of a fibrohistiocytic line of differentiation. Immunohistochemistry showed that distinct lineages were becoming identifiable in these tumours, and the concept of MFH was therefore questioned. So various authors looked at big series of malignant fibrous histiocytomas, and in the early 90s, Dr. Fletcher looked at a large series of so-called MFH and described that these were heterogeneous. These represented a heterogeneous group of tumours, including not only a variety of different sarcoma types, but also melanomas, carcinomas, and lymphomas, unified by a common histologic appearance. So these were, in effect, the common endpoint, morphologic endpoint of a variety of highly malignant pleomorphic neoplasms that had become characteristically more aggressive and had acquired characteristic, much more aggressive genetic events, and all taking on this similar morphology. So in this study, he showed that 60% of MFH were actually definable sarcoma subtypes with an identifiable line of differentiation. 13% were actually non-mesenchymal tumours, for example, sarcomatoid carcinomas, melanomas, and so on. And only a quarter of this series showed no line of differentiation. And even amongst this group, there were no consistent features. And so this was deemed to represent a wastebasket term for a heterogeneous group of unclassifiable tumours, all unified by the presence of pleomorphic morphology. So when we try and diagnose UPS now, some of the difficulties lie in the fact that there are still no fixed criteria for diagnosis, and diagnostic criteria differ between pathologists. And a lot of the time, it depends on really how far we are prepared to go to try and find any line of differentiation, be that with immunohistochemistry or even molecularly. Other diagnostic challenges include, of course, significant tumoral heterogeneity and sampling issues. And we illustrate a case here. So leiomyosarcoma is a tumour that is well known for being morphologically very heterogeneous within the same tumour. And if we look at the same tumour on the left, we have a well-differentiated focus, very discernible as smooth muscle. And on the right, in the same tumour, we have frank pleomorphic leiomyosarcoma with the morphology of undifferentiated pleomorphic sarcoma. So it depends very much on sampling error. And in fact, leiomyosarcomas frequently get upgraded in subsequent excision because core biopsy is not representative. We can see that the tumour on the left would show a French classification score of grade or differentiation score of 1 to 2. The one on the right would show a differentiation score of 3 and also be associated with prominent mitotic activity and focal necrosis. So pleomorphic sarcomas are heterogeneous tumours united by a common morphology. So it's important for us that we are diagnosing, not over calling them, and that they represent a diagnosis of exclusion. So we need to first exclude sarcomas with specific lines of differentiation. So we need to exclude pleomorphic non-mesenchymal tumours, such as sarcomatoid carcinoma. We need to exclude other pleomorphic sarcomas with identifiable lines of differentiation, for example, pleomorphic liposarcoma and pleomorphic leiomyosarcoma, and exclude the expanding category of dedifferentiated tumours. One thing we always need to remember, of course, is that if we see a pleomorphic or spindle cell / sarcomatoid neoplasm in a viscous or organ, this is much more like as a primary neoplasm, not associated with disease anywhere, as an isolated primary neoplasm. This is much more likely to represent sarcomatoid or undifferentiated carcinoma rather than primary sarcoma in that organ. So we can see a pleomorphic breast tumour on the left and the very diffuse and strong keratin expression that this shows. And let's also remember that there are non-specific immunohistochemical findings, such as the presence of myofibroblastic type SMA expression that we can also see in sarcomatoid carcinomas. How undifferentiated does undifferentiated pleomorphic sarcoma have to be? So are we allowed any differentiation at all, or does it show, does our pleomorphic neoplasm just not have to show any discernible line of immunohistochemical differentiation? Well, we're allowed various focal various focal positivity for a collection of markers. So we're allowed to have focal CD34, SMA, desmin, even A13 and CD68. And let's just remember that this should be focal. So CD34 is a very non-specific fibroblastic marker. And if we see focal expression, that's okay. Similarly with SMA and even with desmin, although we are not allowed to have myogenin or MyD1 in nuclei because that makes the tumour pleomorphic rhabdomyosarcoma. Even A13, some tumours can show aberrant expression of A13, but this should be very focal. And if you see it, we should always look at the clinical context to exclude a history of carcinoma, or make sure that the patient does not have carcinoma in a defined organ. This pattern of A13, this is diffuse and strong multifocal expression, is not the kind of expression you would see in as aberrant expression in pleomorphic sarcoma. So we're allowed a little bit of keratin sometimes, but we really have to fully exclude pleomorphic carcinoma, sarcomatoid carcinoma first. And we should not see any sort of widespread expression like this. CD68, again, non-specific marker that we're allowed. So this is SMA expression, subplasmal lemmal accentuation, as Prof was saying, indicating myofibroblastic differentiation, which is relatively non-specific, and we can see a variable amount of that. CD34, non-specific fibroblastic marker amongst a marker for other lines of differentiation, and we're allowed variable CD34. As we said, myogenin and MyD1 expression indicates pleomorphic rhabdomyosarcoma, so we're not allowed myogenin or MyD1. So it depends very much on the type of marker that we are seeing that's positive. In the context of positive desmin, this would make the tumour pleomorphic rhabdomyosarcoma, and this is prognostically important because pleomorphic rhabdomyosarcoma is one of the much more aggressive pleomorphic sarcomatous neoplasms. Let's now move on to mesenchymal tumours that can have pleomorphic sarcomatous morphology. So pleomorphic liposarcoma, we're clear cut with that. So that's pleomorphic sarcoma with atypical lipoblast. Sometimes they can be focal, so we can miss it on core biopsy. Dedifferentiated liposarcoma, we have just talked about this. This is pleomorphic non-lipogenic sarcoma arising from well-differentiated liposarcoma. Most retroperitoneal pleomorphic sarcomas, undifferentiated, are dedifferentiated liposarcomas with MDM2 amplification. Precise characterization, as we mentioned, is important because of targeted therapies to dedifferentiated liposarcomas, which would not work in other pleomorphic sarcomas. Pleomorphic leiomyosarcoma is another tumour that can manifest with markedly pleomorphic morphology. Pleomorphic rhabdo with variable myogenin and MyD1 expression. Malignant peripheral nerve sheath tumour can manifest as a spindle cell, atypical spindle cell, or pleomorphic neoplasm. Let's remember that, as Prof mentioned, S100 protein is seen in only about 50 to 60%, and it is typically very focal. Yeah, not diffuse, just very focal. Osteosarcoma can also have pleomorphic sarcomatous morphology. So the other mimic of undifferentiated pleomorphic sarcoma is the group of the expanding group of dedifferentiated tumours. Dedifferentiation occurs in a subset of tumours in which there is high-grade tumour, anaplastic tumour, without evidence of the line of differentiation of the original beta-differentiated neoplasm. So we can see an example of chondrosarcoma, so well-differentiated chondrosarcoma on the left, and this abrupt demarcation with dedifferentiated chondrosarcoma on the right. Dedifferentiated tumours typically manifest with pleomorphic morphology. So with the morphology of undifferentiated pleomorphic sarcoma, we can see a dedifferentiated GIST on the left, dedifferentiated liposarcoma on the right. So most of us are aware that there are a variety of tumours that can dedifferentiate. So established ones include chondrosarcoma and chordoma. Liposarcoma, we know that solitary fibrous tumour can rarely dedifferentiate, as can dermatofibrosarcoma. An emerging group of tumours that are now known to be able to dedifferentiate are the group of GISTs, which we'll talk about. And let's also remember that carcinomas, including those of the salivary glands, can also dedifferentiate, as can melanomas. And that's something that a lot of us can forget. So dedifferentiated tumours can arise de novo or as a complication of recurrent previously well-differentiated tumour. So about 10% of well-differentiated liposarcomas dedifferentiate, particularly in deep sites where they're not easily clinically monitored. And there is typically this very abrupt demarcation or transition between the well and the dedifferentiated components. Dedifferentiation invariably confers a much more aggressive behaviour. It's an example of dedifferentiated solitary fibrous tumour in which there is typical, just very bland, classical solitary fibrous tumour on the left, and then dedifferentiation on the right. So genetically, dedifferentiated tumours have the same underlying genetic mechanisms as their well-differentiated counterparts, but they acquire other genetic events that drive tumour progression and biologically aggressive behaviour. So as we mentioned before, dedifferentiated liposarcoma also has MDM2 amplification and other amplifications in the 12q13 to 15 amplicon, but acquires additional genetic events, including amplifications at ASK1 and JUN genes. We need to be aware of dedifferentiated tumours because of clinical implications. They have distinct genetic backgrounds and therefore there may be potential for targeted therapies in the future. So a mention on dedifferentiated GIST, and that's because it's a rare entity that is more emerging. Probably a lot of us have not thought to diagnose it. Maybe it's been underdiagnosed because it has been diagnosed as sarcomatoid carcinoma or dedifferentiated liposarcoma or pleomorphic sarcoma NOS. But these are increasingly being described. And as we are aware, GISTs can undergo progressive atypia, morphologic atypia, with chronic imatinib treatment. And patients who have dedifferentiated GISTs frequently have a long previous history of GIST with chronic imatinib treatment. However, dedifferentiation in GIST can also occur de novo. These show altered morphology, typically presenting as undifferentiated pleomorphic sarcomas, and they lose CD117 expression. Dedifferentiation in GIST has mostly been described in the stomach, but has also been documented at every site in the GI tract. As expected, these occur in older adults, and most of them show normal areas of conventional GIST, so either spindle cell or epithelioid GIST. And these tumours show morphologic features predominantly of undifferentiated pleomorphic sarcoma. There can be some variants, so some tumours undergo rhabdomyosarcomatous differentiation, some can show epithelioid or pseudopapillary morphology, and one case has shown angiosarcomatous differentiation. These are high-grade malignant tumours associated with prominent necrosis and mitotic activity. So these are invariably associated with loss of CD117, and frequently these tumours show loss of CD34 expression. Two caveats, so they can, for reasons that are unclear, show gain of pankeratin. Very strong expression is seen in this dedifferentiated GIST, which means that we might overcall this, we may aberrantly diagnose this as sarcomatoid carcinoma, and they can also gain desmin expression. Mutational analysis will show an underlying KIT or PDGFRA mutation, which is the same as the original genetic event. So the same mutation as was noted in the original spindle cell or epithelioid gist. And there is no difference in KIT genotype between conventional and dedifferentiated components, although there may be the acquisition of further mutations in KIT or PDGFR. And dedifferentiation in GIST is thought to be secondary to genomic instability. There has been authors have shown loss of heterozygosity of KIT or low-level amplification of KIT in small numbers of cases, implying that these are events that are secondary to the increasing genomic instability of this neoplasm. So for us to remember that dedifferentiated GIST is in our differential diagnosis of pleomorphic neoplasms, so pleomorphic sarcoma-like neoplasms in the GI tract or abdominal cavity. Let's not overcall them or call them pleomorphic sarcomas, or because they frequently can show strong AE13 or other cytokeratin expression as sarcomatoid or pleomorphic carcinomas. Dedifferentiated melanoma, I thought we'd talk about because we are seeing more of these coming in as undifferentiated pleomorphic sarcomas, and it's an evolving concept. And I think that, again, like GIST, many of us may have undercalled them in the past because of their sarcoma-like morphology. So this is a relatively newly defined concept, which can occur in primary or metastatic melanoma. So the term dedifferentiated melanoma refers to the transition between conventional type melanoma of any morphology and the undifferentiated component, whereas undifferentiated melanoma lacks histologic or immunohistochemical evidence of any conventional melanoma. Again, as with other dedifferentiated tumours, there is frequently abrupt demarcation. The undifferentiated component typically has the morphology of undifferentiated sarcoma, and these components, dedifferentiated or undifferentiated melanoma, show loss of melanocytic markers, including SOX10, so including highly sensitive melanocytic markers. All these are lost. However, they may gain markers suggesting other pleomorphic neoplasms. So there can be gain of CD10 and P63, which may lead us to an aberrant diagnosis of pleomorphic dermal sarcoma or sarcomatoid carcinoma, respectively. So this highlights the importance of clinical correlation, particularly in pleomorphic or undifferentiated neoplasms in nodal sites, because let's remember that dedifferentiated melanomas just look like sarcomas and show complete loss of melanocytic markers, even the most sensitive ones such as SOX10. So we need to be aware of this diagnosis and look for areas of better differentiated melanoma, including the appearance of nesting and focal melanocytic marker expression. So these are the pointers towards a diagnosis of dedifferentiated or undifferentiated melanoma. So of course, clinical correlation is everything. So we need to be aware of any previous history of melanoma, be able to look for and be aware of having to look for small areas of differentiated melanoma. And that might be tumor nesting or more epithelioid cells with prominent nucleolation. If we see undifferentiated histology not fitting with any specific entity, if we are encountering tumor at a site that is unusual for primary undifferentiated pleomorphic sarcoma, such as in the axilla, in the neck, in the inguinal area, or in the GI tract. So at nodal sites or some visceral sites. Also, if we see an unusual multifocal disease pattern, typical of the spread of melanoma, so widespread metastases that we do not typically associate with the pattern of progression of undifferentiated pleomorphic sarcoma. Also, if we see a genetic mutation in keeping with melanoma, and we've had a few cases now where we have had to rely on the presence of BRAF or NRAS positive mutation, to diagnose these as melanomas because all markers were negative. Also, the absence of another primary, such as anaplastic carcinoma, in other organs. So moving back to pleomorphic sarcoma, after we've excluded all the mimics, let's talk about the genetics. So there are no useful genetic tests that we can use in the diagnostic setting because there are no recurrent genetic abnormalities. These tumours, pleomorphic sarcomas in general, are associated with complex, unbalanced karyotypes as a function of marked genomic instability. And various studies have shown that these tumours have structural rearrangements and copy number shifts of almost all chromosomes. So there are no recurrent events and nothing that we can use in the diagnostic setting. There are some tumor-associated amplicons, such as amplification of YAP1 or VGLL3. And often these tumours are the targets of frequent deletions in tumour suppressor genes such as P10, P16, RB1, and P53. There are also various telomeric aberrations. So behaviour, 75% of UPS tumours have been defined as highly aggressive. And we know it's a heterogeneous group of tumours, but we wonder why not 100% of them have been aggressive. Well, in recent years, there has been the much more widespread availability of molecular techniques, including massively parallel sequencing studies. And with the results of these studies and even more pronounced clinicopathological correlation, authors have been able to distinguish pleomorphic sarcoma-like tumours with much more indolent behaviour. So these tumours have the morphology of pleomorphic sarcoma but behave in an indolent manner. And Prof has talked about these. I'll be quick with them, but just to highlight previous tumours that probably have been overcalled as undifferentiated pleomorphic sarcomas, but in fact show much more indolent clinical behaviour. So the first of these is the superficial CD34 positive fibroblastic tumour, which occurs in young to middle-aged adults and is known for its indolent behaviour. These tumours have not been shown to metastasize. So these are typically circumscribed superficial, i.e. subcutaneous tumours in which there are variable but generally marked and bizarre nuclear features, including intranuclear inclusions. In contrast to undifferentiated pleomorphic sarcomas, these have extremely low mitotic activity and usually no necrosis. The cellularity is typically, if you really observe these, is really only moderate. The marked atypia can be very confusing with these pleomorphic, bizarre cells, but these are more scattered in fibrous stroma, and they have extremely low mitotic index. Interestingly, also, they invariably show diffuse and strong CD34 expression and frequently show focal pancytokeratin expression. So that's the pattern of CD34 expression. The other tumour, again mentioned by Prof, is the PRDM10 rearranged soft tissue tumour. So this has been identified in a small subset of tumours previously called MFH, and again briefly have shown incredibly indolent behaviour. Similar to superficial CD34 positive fibroblastic tumours, these have extremely low mitotic indices in contrast to the degree of pleomorphism, and have shown no metastatic behaviour, very rare local recurrences. And similar to the previous tumour, these show invariable CD34 expression and focal keratin expression in most of them. If you have PRDM10 immuno, this is also positive. So let's move on to intimal sarcoma. So some brief words on updated aspects of intimal sarcoma. So these are malignant mesenchymal tumours arising in large vessels of the systemic and pulmonary circulations and the heart. They're characterised by intraluminal growth with obstruction of the lumen and frequently emboli to peripheral organs. They typically occur in proximal vessels. 80% involve the pulmonary trunk, with up to 70% involving right or left pulmonary arteries, 40% involving both. And cardiac involvement, frequent cardiac involvement, in fact, they comprise the most frequent cardiac sarcoma type. So 40% of cardiac sarcomas are intimal sarcomas. 40% show either metastatic tumour or tumour to the lungs or direct infiltration of the lungs, and extrathoracic spread occurs in about 20%. More rarely, they can involve the aorta, predominantly the abdominal aorta, with a smaller number in the thoracic aorta. Genetically, these are heterogeneous, but they have been associated with recurrent amplifications in, again, the 12q12 to 15 locus, similar to dedifferentiated and well-differentiated liposarcoma, which includes a variety of oncogenes, including CDK4 and MDM2. And these have also been associated with PDGFRA, KIT, and EGFR amplifications, along with gains and losses at other sites. So these tumours have shown high-level amplifications in PDGFRA and or EGFR in 75 to 80%, and 2/3 of these have shown high-level MDM2 amplification. So that's helpful in establishing a diagnosis, although 1/3 of these do not show MDM2 amplification. These have a non-distinctive and very heterogeneous morphology, typically manifesting as pleomorphic sarcomas, NOS, not otherwise specified, and have a variety of morphologic patterns, but are high-grade pleomorphic sarcomas. Sometimes they can be more spindled and MPNST-like, and they can show variable heterologous differentiation, including heterologous osteoid and chondroid. Next tumour is ossifying fibromyxoid tumour, which I've included because it's difficult to pigeonhole in any other of the lectures, and also because it's rare and it has quite an unusual look to it with a spectrum of morphologies. I thought we'd include it so that we can get an idea of the basic morphology, the defining morphology. So this is a rare mesenchymal neoplasm of uncertain differentiation, characterized by cords and trabeculae of ovoid cells in fibromyxoid matrix. And this is often, but not always, surrounded by a shell of lamellar bone, which may be complete or incomplete. These are tumours with the potential for recurrence and metastasis to visceral and soft tissue sites, but particularly when they show histologically malignant features. They can occur at any site, frequently in the subcutis, but 40% occur in the thigh. They can occur in skeletal muscle of the extremities, but also in head and neck sites and in the trunk, and have a wide age distribution, but most frequently occur in middle-aged adults with a male preponderance. Malignant OFMT is characterized by high nuclear grade or high cellularity and the presence of more than 2 mitotic figures per 10 HPF. So morphologically, as we mentioned, these tumours have a complete or incomplete shell of lamellar bone, but this can be lacking, particularly on core biopsy. So we need to know that this tumour exists, and we need to know how to look for the morphology without the presence of bone. In this picture on the right, we can see that the bone actually impinges into the substance of the tumour, dividing it into cellular lobules. So the distribution of the bony elements can really vary. On the picture on the left, we can see that there is an incomplete shell of mature bone. On the right, there is no bone, but the tumour has a lobulated architecture with the surrounding fibrous tissue. So we can see the morphology here, which is very distinct. Very bland. This is the translocation associated neoplasm. So it's composed of very bland ovoid cells, very uniform looking in cords individually and in nests in this prominent, delicately collagenous matrix. Not typically associated with mitotic activity, no atypia or necrosis in normal forms. We can see a more asana-like distribution on the left here. Very cord-like appearance, disposed in extensive, delicately collagenous stroma on the left. And then we can see these cords, tightly packed cords and nests in variable collagenous stroma on the right. Sometimes the morphology can differ. On the left, we can see prominent epithelioid-looking cells, very plump cells with clear cytoplasm. And on the right, even though the cells are disposed in cords, they are somewhat spindled. So ossifying fibromyxoid tumour has a non-specific immunoprofile, and we need to be aware that it may variably express these markers here. So characteristically, almost 2/3 express S100 protein. This can be diffuse in the picture that we can see here, or it can be more focal. But we can see these very bland cords of cells with strong S100 protein in a diffuse pattern here, but it can be more limited. 50% show GFAP expression. Again, all of this is variable and frequently focal. 10% show variable keratin. Small numbers can show variable SMA and desmin. And mp4 can be expressed in a third of cases. And interestingly, there can be INI1 or SMARCB1 mosaic pattern loss in 3/4. Genetically, at least 85% have a gene fusion, so these are fusion-associated neoplasms with balanced gene fusions, leading to very bland morphology. 50% of these show PHF1 fusions, which can partner with a variety of partners, particularly EP400. So 50% of these tumours have PHF1 fusions. There can be rare fusions instead with the B-core genes and other genes. Let's move on to extraskeletal myxoid chondrosarcoma. So again, it's a tumour that most of us are quite familiar with the morphology, but just to explain the genetics and some variant morphologies. So this is a malignant mesenchymal neoplasm of uncertain differentiation characterized by very prominent multilobulation of cells in extremely uniform cells in cords, strands, and nests in a very prominent myxoid matrix. So lobulated tumours are very bland cords and nests of cells in a prominent myxoid matrix. It's a fusion sarcoma characterized by NR4A3 gene rearrangements, and these do not show actual chondroid. So extraskeletal chondrosarcomas. What are the types of them? We can divide them into 3 types. Well differentiated, which can dedifferentiate, which is simple enough. Mesenchymal chondrosarcoma, which is around an ovoid cell neoplasm with these tightly packed uniform cells with variable islands of hyaline cartilage. Again, this is prominent. This is actual chondroid, and then finally extraskeletal myxoid chondrosarcoma, which does not actually show chondroid and is a translocation associated neoplasm, but shows very prominent myxoid stroma, somewhat resembling myxoid chondroid elements. These occur in adults, typically older than 35 years, with an equal sex distribution, and typically occur in deep sites, particularly the lower extremities, but they can occur at a variety of sites, including the head and neck, including rarely in intracavitary sites and extremely rarely in the bone. These are clinically typically slowly growing tumours, which can exceed large sizes and are variably aggressive. These are noted for the ability to metastasize quite late after 1 or even more decades, including to the lungs. And often there is prolonged survival, although there are high rates of distant recurrence and disease-related death. So morphology, we can see these bland cord-like distributions of cells with no atypia, very prominent myxoid morphology, with these myxoid lobules separated by fibrocepta, extremely non-atypical cells. Again, this is another tumour with a non-specific immunoprofile, and immunohistochemistry is essentially not helpful other than the fact that it is negative for everything in contrast to the tumours in its differential diagnosis. So it can variably have expression of any of these markers, but if we do have expression, it's typically very focal and quite weak. So S100 protein can be variably positive. Usually it's focal and relatively weak if it's there. SOX10 is invariably negative. There can be variable EMA, ALK, neuroendocrine marker expression, but again, let's remember that this is generally very weak and focal. We are not expecting to see diffuse and strong or multifocal expression of any of these markers. Again, this highlights the variable positivity and the negativity. So it's negative for myeloid markers, CD34, CD31, and it's always negative for SOX10. So just remember, this has a lobular architecture and a hypovascular stroma, but let's also remember that in rare cases, the tumours can be hypercellular, and particularly on core biopsy, this can be really confusing because it does not have the characteristic architecture of EMC. So we can see a very solid tumour here of typical bland cells, and if we look carefully, we can see that there are small myxoid pools. On the left is a tumour with extensive cellularity, and on the right we can see lots of cellular areas with focal myxoid areas with reticulated networks of similar cells. So we need to remember that there can be cellular variants, or on core biopsy we may only be seeing the cellular focus of tumour. The stroma is very hypovascular, and there may be small microhemorrhages. Again, more cellular components on the right, but in the right of that field we can see the classical morphology. So EMC is defined by fusions of NR4A3, and this gene fuses with a variety of partners, but most frequently EWSR1. So 2/3 of EMC are show fusion of NR4A3 with EWSR1. It can also fuse with a variety of other genes. And some authors have shown that EMC with non-EWSR1 fusions show a higher instance of rhabdoid immunophenotype, of high-grade morphology, and may be associated with a more aggressive outcome than EWSR1 NR4A3 positive tumours that are the most common types. Key diagnostic points: these are multinodular deep tumours with very bland cords and nests of cells, with myxoid stroma, which is very hypovascular but may have various hemorrhage or microhemorrhages, non-specific immunoprofile, typically negative for everything, and is associated with specific NR4A3 gene fusions. Differential diagnosis is wide and contains contains these diagnoses. So mucinous carcinoma and chordoma should be relatively straightforward to separate with immuno, because these two entities will show strong and diffuse keratin expression, or for mucinous carcinoma, and strong and diffuse keratin EMA and S100 protein expression, typically in chordoma, in contrast to the absence of expression or only weak focal expression of these markers for EMC. Myxoid liposarcoma can be in the differential diagnosis, but I think we are aware of the morphology of myxoid liposarcoma with its very prominent curvilinear vascular pattern, in contrast to the hypovascular stroma of EMC. Myxofibrosarcoma has variable atypia, doesn't have a cord-like cell network, and again, it typically shows prominent, this time large, thin-walled vessels, in contrast to the hypovascular myxoid stroma of EMC. Primary pulmonary myxoid sarcoma is a tumour that typically occurs in females in the thorax, and is associated with EWS-CREB fusion. We've talked about ossifying fibromyxoid tumour, which is a genetically distinct entity, and myoepithelial tumours can show a variety of morphologic appearances, but can sometimes show reticular distributions in myxoid stroma. And we come to the end of this talk now. So thank you.